my name is Dr. Charlie Hindmarch and I'm a genomics researcher at the Queen's Cardiopulmonary Unit, or QCPU. QCPU provides state-of-the-art research infrastructure and expertise to the Queen's community. Founded by Dr. Stephen Archer, a MEDS 81 graduate, QCPU provides 8,000 square foot of research infrastructure to the Queen's community, which offers a bench-to-bedside translational research approach. QCPU is located in the heart of Queen's campus, next door to the medical school and opposite Summer Hill. So at QCPU, we primarily focus on heart, lung, blood and vascular research. But we also provide research infrastructure across the university. So if you're a cancer researcher or if you're in bioengineering, we probably have some of the tools to help you achieve your research ambitions. In 2020, we helped 3 faculties, 16 departments and 27 research groups. And since 2019, we've trained 73 scientists. So QCPU has 2 main arms of research, the first being clinical research. But the basic scientific research is split between 5 research laboratories, who are staffed by 5 highly qualified scientists who have been recruited from around the world. So today, I will take you on a journey from populations to cells to genetics. And I'm going to introduce you to the scientists who are going to tell you about the infrastructure that they have in their laboratories and how they address the vital challenges we face today. QCPU is a translational research facility, which means it's particularly important that we have access to a clinic. And what this does is actually this takes patients from the Kingston community and offers echocardiography services for part of their normal clinical care. One advantage to having sonographers and registrars and medical staff on site is that we can also use these facilities in order to be able to facilitate active research projects and translate the findings in the laboratory back into the clinic and back to the bedside. So the first lab we're going to go to today is our histology lab, and I'd like to introduce you to Oliver Jones, who is a clinical histologist who is trained in the United Kingdom as well.
Hi, I'm Oliver Jones. I'm the histologist here in the QCPU. So I run the histology service. So what is histology actually? So histology is simply the study of tissue. So what that means is we take tissue from either clinical samples or from research samples, and then we process them so we can look at them down the microscope to see at the cellular level what's going on with the tissue. Now, as you can imagine, if you took a piece of tissue and tried to cut it to put on a microscope, it's going to move around quite a lot. So to do that, we have to process it into a hard medium, which we use wax, so we can cut the sections to put on the microscope slide. So here is our tissue processor. Now this processor will take the tissue, which comes to us in a fixative to preserve the tissue and to kill any bacteria or viruses. And it takes it from the fixed state into a state where we can put it into wax. So over here is our tissue embedding station. And what this does is it takes the wax and it puts it into mold, and we can embed the tissue inside that wax, and then we cool it down to give a hard block. So here now we have a block of tissue that is embedded in the wax. Now we've cooled it on this cold plate so that it makes the wax more hard. And the hard wax enables us to cut an ultra thin section. So when I say thin, we cut it at 4 microns, which is 4/1000th of a millimeter. So we'll cut a section and we'll try and cut a ribbon of them, which will then float onto water and pick up on the microscope slide. I'm cutting the sections and I'm cutting a ribbon so we can have multiple sections. Now as I'm cutting it, I actually gently blow on it, which helps the section to come off smoothly. Then we're going to lift the ribbon and float it on the warm water bath. Now what this does is it helps the tissue to just spread out and makes it nice and smooth for when we look down the microscope. As you can imagine, any small defect or artifact will be magnified as soon as you put it under the microscope. But when we have the nice ribbon of sections spread on the nice warm water bath, we can pick them up on the glass slide and then they can be ready for staining to look at down the microscope. So when we first pick up the section, this is how it looks. You can't really see anything, it's all white and pale. Now to see it down the microscope, we have to add some colours, so we do some special stains, and this is called an H&E or hematoxylin and niacin stain. And this is how it looks, and then we can look at this down the microscope. So another service that we can provide in histology is the electron microscopy. So the difference with electron microscopy and light microscopy is the resolution that we can get to. Typically on a light microscope you can get to 400 times magnification, but on an electron microscope you can go way further down. You can get into the hundreds of thousands times magnification, and with that we can look at the ultrastructures within the cell, such as mitochondria, or we can also look at viruses such as COVID.
So welcome to Proteomics at QCPU. I'm standing in front of a state-of-the-art, world-class piece of infrastructure that allows us to measure multiple proteins simultaneously within hundreds of thousands of cells. And what this allows us to do is give us a really clear advantage in terms of being able to describe how different cells within your blood or within certain tissues behave. So you can see here, this is the sample actually running. These crosshair is a laser that's drawing across and actually burning the tissue. And as it's burning the tissue, you can see it's actually creating this image here, which shows us the different cell types within a tissue. So now we're in the cell culture and cytometry lab with Curtis, who's going to show you around.
Hi, my name is Curtis Noordhof. I'm a research assistant here at QCPU, and I work with Dr. Patricia Lima. We are QCPU's molecular imaging specialists. The services that I offer here are cell culture, flow cytometry and sorting, and confocal microscopy. On this walkthrough, I will give you a quick overview of what I do and show you some of the equipment that I work with. Cell culture is the process by which cells are grown under controlled conditions, generally outside their natural environment. After the cells of interest have been isolated from living tissue, they can subsequently be maintained under careful, controlled conditions. We use an inverted microscope to observe if the cells are growing well and ready for treatment, or if they require more time to grow. We need to stimulate their growing conditions, so we place our flask in these incubators. These incubators are set to 37 degrees and 5% carbon dioxide to create the most ideal growing environment. Confocal microscopy, similar to fluorescent microscopy, allows one to detect the presence and localization of fluorescent molecules in a sample. The main difference between a confocal microscope and a standard fluorescent microscope is the presence of a pinhole that allows one to exclude photons coming from a different focal plane. The confocal microscope is a specific fluorescent microscope that allows obtaining 3D images of the sample with good resolution. This allows to reconstruct a 3D image of the sample. Flow cytometry is a technique used to detect and measure physical and chemical characteristics of a population of cells or particles. In this process, a sample containing cells or particles is suspended in a fluid and injected into a flow cytometer instrument. The sample is focused to ideally flow one cell at a time through a laser beam, where the light scattered is characteristic to the cell and their components. Cells are often labeled with fluorescent markers, so light is absorbed and then emitted in a band of wavelengths. Tens of thousands of cells can be quickly examined, and the data gathered are processed by a computer. Flow cytometry is routinely used in basic research, clinical practice, and in clinical trials.
Okay, so welcome to my lab, which is Genomics, Transcriptomics, and Molecular Medicine. And what we do in this lab really is we try and read the book of life. So we know that within every single cell of your body, you have DNA. And what that DNA does is that DNA encodes proteins. So it literally gives a recipe of instructions to tell the cell how to behave under different circumstances. Now, the trouble with reading the genome is that the human genome is really big. It's about 3 billion letters long in an alphabet that just has 4 different letters. And of these 3 billion different letters, if I was to read them out 1 letter per second, 24 hours a day, 7 days a week, it would take you over 100 years to be able to read that book out. And you still wouldn't understand what the book meant. And that's very much what we do in this lab. We try and read that code and look for spelling differences that exist between people who have diseases and people who don't have diseases. We also look for individual words or genes that might be expressed more than others or less than others. And by doing this comparison, we can really drive discovery forward, because we can start saying, in this disease circumstance, these people have a particular spelling difference that might contribute towards the reason why they've got the disease in the first place. Or maybe they make too much of a protein and that's encoded at the DNA level. So it's incredibly important that in this lab, we're matching up wet laboratory skills, we're using pipettes and we're using equipment around us like this, but also to be able to understand that data, we're using state-of-the-art computational skills to be able to take these small fragments, glue them back together into a genome so that we can start to understand this book. So now we're at the preclinical and nuclear medicine lab with Elahe. Let's go and take a look around.
Hi, my name is Elahe Alizadeh. I'm a radiation physics scientist conducting nuclear imaging laboratory at QCPU. This laboratory contains different sections that I will introduce you. The first one is animal preparations for anesthesia, surgery, and animal monitoring. This is the section for handling radioactive materials with considering radiation safety protocols properly, especially working with TLC scanner for doing labeling of antibodies and proteins with radioactive materials and doing quality control before injecting radioactive materials to small animals. The main equipment in the lab is tri-modality nuclear imaging system from MI Labs. It's a CT, PET, and SPECT scanner equipped to anesthesia systems and gating systems for gated imaging. Also equipped with computers for image reconstructions and image processing. What we have here in this lab is tri-modality CT, PET, and SPECT together. As I said, multimodality techniques help us to get complementary information about the subject and help us to overcome the limitation of each technique. But the application of this multimodality technique is not just cancer study. We can use this equipment for other type of studies like anatomical and functional studies in cardiopulmonary models. And this is the same image using contrast agent Electron with the same CT database, but showing the beating heart that we call it 4D imaging. This one is an example of gated SPECT in a rat model developed to study pulmonary arterial hypertension. These are some examples of ex vivo imaging with very high resolutions. So ex vivo imaging means that the organs are not inside the live animals, but we use contrast agent to discriminate very soft and small vessels in lungs and heart. Some other applications are ex vivo high resolution CT of bone morphology, control rat, and in a rat with chronic kidney disease mineral bone disorder. I'm explaining CT scanners for kids, maybe good examples to show application of CT scanners is using Kinder Surprise. Anything you have at home can be scanned by a CT scanner.
So thank you very much for coming on the walkthrough of QCPU with me. I hope you found it interesting, and if you'd like any more information, you can contact us at qcpu@queensu.ca.